everyone to today's session of the AI for Net Zero webinar series. My name is Amal Sheikh, and I will be co-chairing today's session with Minjia Zhu. And today we are having two early research career presenters. As a background, the AI for Net Zero webinar series is about topics which are at the intersection of artificial intelligence and various domains which are crucial to achieving net zero objectives, including energy, transport, environment, agricultural, and food systems. Today, we have two different speakers from two different domains. We will start with Alhasan. Alhasan is a postdoctoral research associate at Harvard University, and he works on the EcoAI project. His research is focused on deep learning at the intersection of advanced machine learning algorithms and subsurface simulation. He worked in geological modeling using generative modeling, and he's currently working on subsurface simulations at the full scale using deep learning process. He obtained his PhD in 2023 in the geoenergy engineering, focusing on geostatistical simulation using generative AI. Alhasan, before that, he had a master's in engineering mathematics from the Polytechnic University of Catalonia in 2019, and his BSc in communication and information engineering in 2018 from the University of Science and Technology at Zuwel City. He is generally interested in the topics of scientific computing and machine learning and AI. Alhasan, you are first, so the floor is yours.
Right. Thanks a lot, Ahmed, for the introduction. So my talk today is about generalizing generative adversarial networks for subsurface geomodeling. So this work was funded by Total Energies as a part of my PhD in Harvard University. So let me start by talking about the key steps in geostatistical simulation. So here we have a training image that describes conceptual geology. So for example, this is the solid phase, and the yellow bars here are the channel phase, which could describe a fluid that passes through. So this image is used to train a geostatistical model to generate multiple realizations that are similar to the training image. The next step is to condition this realization on some observed data from a particular subsurface reservoir. This data could be soft data like seismic surveys, for example, or hard data like well logs. So the conditioned realizations should respect both the training concept and also the observed data. The third step is to perform some analysis on these conditioned realizations. So for example, we can do some uncertainty quantification on a particular simulation problem, or perhaps solving an inverse problem. But it is often the case that the conditioned distribution is different than the training image. And this raises a question of how good the conditioning algorithm can be. So this is a well-known concept in machine learning called model generalization, which is the ability of the model to perform well on unseen patterns. In terms of geostatistical simulation context, this means the ability of the model to condition on unseen observed data. But the discrepancy between the conditioning distribution and the training distribution could be due to a bias in the data. So there could be some certain assumptions in the training concept which don't necessarily apply for a particular subsurface reservoir, or the data itself could be limited in terms of the variability of the geological phases. So these challenges could hinder the model ability to condition properly on the observed data. So the question that we tried to answer in this project was, can we improve the generalization of the model if we are faced or presented with these challenges? And we try to answer this question both in a global conditioning context and also a spatial conditioning context. So the first part which deals with the conditioning on global properties, we selected the global proportions of geological phases. So, for example, on the left side here, you can see a binary phases data set. So we have the solid phase, we have the channel phase, and in this case, we have three different channel proportions. So the proportion is just the percentage of the yellow part in the image. So we have here 25%, 30%, and 35%. And the question is, can we generate like geologically consistent images, but with different global proportions, like for example, 27% or 32%, or even beyond this range, like 20% or 40%. So this is the first data set. The second data set was a multiple phases data set. So in this case, we were interested in generating different display proportions, the blue regions here. So here we have like two different classes of proportions, 4% and 7%. And again, the question was, can we generate something that's different, maybe like 5 or 6 or 2 or 8 percentages? So this is the first part, which is conditioning on global properties. The second part is to condition the realizations on soft maps. Soft maps are basically probability maps that describes the spatial distribution of the geological phases. So you can see, for example, here on the left side, we have artificial samples, obviously, and there is a trend of decreasing the channel widths from left to right. On the right side here, we have binary masks of Braham Porter River. So these are real samples. And again, the question was, can we generate something that's different from the training set? Maybe high proportions on the right side or in the middle. And you can also see that these pattern on the left side here are non-stationary, because there is this kind of trend of a decrease in the channel widths. On the other hand, for the real samples, these could be non-stationary or non-stationary or stationary because they are coming from a realistic scenario. So the method that we use to generate these realizations is called Generative Adversarial Networks, or GANs. These are neural networks that can be trained to learn the distribution of a particular data set. And once we train these models, we can generate new samples that resemble those found in the training set. So the basic architecture of GANs consists of two neural networks, the generator and the discriminator. Both of them could be convolutional neural networks. The generator maps noise, random noise, to the pixel space, to a generated image. The discriminator, on the other hand, takes both real samples and the generated samples and tries to distinguish between them. So it outputs a probability of the sample being real, that's coming from the training samples. So it's basically a binary classifier, and it is optimized to minimize the classification loss. On the other hand, the generator is optimized to maximize this loss so that it can push the generated samples toward the real distributions. So this defines a min-max optimization problem between both the generator and the discriminator. This optimization problem goes iteratively until the generator is able to generate samples that are indistinguishable from the training set. But so far, the conditioning is purely stochastic. There is no way to force the generated sample to respect any form of conditions or properties. To do that, we used a variant of GANs called conditional GANs, and the main difference is we can pass some auxiliary information to both the generator and the discriminator. This auxiliary information Y can hold specific information about the dataset, for example, just the global proportion. So if we pass the global proportion to the generator, we expect the generated samples to generate realizations that respect this global proportion. For the discriminator, in this case, it outputs a conditional probability that describes not just how realistic X is, but also how good it correlates with the condition Y. So the question here is how to pass this condition to the generator. So in the case of global proportions, we tried different methods for this kind of conditioning. The naive approach is just to concatenate both the stochastic vector Z and X. Another approach is to use what's called a conditional batch normalization method. This method tries to modulate the activations of the normalization layer using this condition. And finally, we proposed another method which is based on the conditional batch normalization, but the only difference is that the modulation or the conditioning happens only on the shifting parameter of the normalization, while the scaling parameter is kept fixed. And you can see here, when we compare these methods on both datasets, the channels dataset and displays dataset, the outlier percentage of the proposed method is smaller than the other two methods. We were also interested in the way we sample the conditions during training. So the mainstream method is just to sample the condition in a discrete way, which is the condition presented in the training set, like the 25%, 30%, and 25 and 35% are sampled discretely during training. We thought of a method that we can actually sample everything that we want the model to generate, whether these conditions presented in the training set or not presented in the training set, and we call this a continuous sampling. And you can also observe here that continuous sampling has achieved a much lower outlier percentage when compared to the discrete sampling. So these are some results for the binary phases data set. And you can see here that these are generated just by fixing the noise vector and gradually increase the condition Y from 25% to 35%. So again, the condition Y represents the global proportions of the channel here. So just by gradually increasing Y, you can observe that new channels starts to appear, and the whole global proportions increases gradually, as you can see also in this chart here. In this histogram, we just plotted the represented conditions versus the unrepresented conditions. So the represented means that these were found in the training set. The unrepresented means that these weren't in the training set. And you can see that the model were able to generate something beyond the training samples. We also use some kind of correlation functions to assess the geological consistency of the generated samples. So we use two functions here, the 2-point probability function and the 2-point connectivity function. So the 2-point probability function tells us the probability of finding 2 points in the spatial domain separated by a specific distance that belong to the same phases. And you can see here that when we increase the target proportion from 25% to 35%, this probability increases, which makes sense because it's more probable to find 2 points in space that belongs to the channel phase when we have larger proportions. The same also holds for the connectivity function, except in this case, it's more likely to find 2 points that are connected together. But we can also see the same kind of trend when we increase the target proportions from 25% to 35%. We have a similar figure here for the multiple phases dataset. Again, the condition, the noise vector is fixed, and the condition y, in this case, represents display proportions, the blue regions here. And you can see that when we increase the condition from 4% to 7%, the display proportions globally gradually increases in the image. You can also see in the chart here, it's the increase is almost linear as between the generated proportions and the target proportions. The second problem that we were interested in is to condition on spatial maps. So in this case, the condition is not just a scalar value, it's actually a map. This map describes the spatial distributions of the geological phases. And for this problem, we use the method called SPADE, or spatially adaptive denormalization. And it is very similar to conditional batch normalization, except that the scaling and shifting parameters are calculated per pixel instead of per the entire domain. And this way, it can be used to modulate each pixel independently. So the conditioning maps are passed to 2 successive convolutional layers to calculate the scaling and shifting parameters, which are used to modulate the normalized activation here. And these are some results on the artificial data set. So here, we pass the condition y to the generator, and in every sample here, it is generated with a different z vector. So you can see that all 4 samples respect the given condition, while each one of them is different, is stochastically different than the others. And here I'm just plotting the mean map across the whole spatial domain. So this is calculated. The mean is calculated across 50 generated realizations. Then we can generate something that is totally different from the training set. For example, we want to have like high channel proportions on this part, while there is no channel at all, so these parts are 0, and these are low channel proportions. So we can see that the model is able to generate something that respects the conditioning map while also being geologically consistent. And these are the results on the real masks. So these are real scenarios, and this is just purely random conditioning map. So doesn't have to be in the training set. You can see that it is designed to have this kind of direction of the channels. And again, the generated realization respected this conditioning map while being stochastically different than each other. And again, this is just a different result. Sorry. Yeah, this is just a different result. In this case, the direction is in the opposite side. And again, the generated realization respected this given condition. So with this, I'll conclude my presentation with using the modulating algorithms like conditional patch normalization or SPADE allowed us to allow the generative model to learn the correlation between the data points and the conditions in an implicit way. So we didn't have to force this learning with a specific loss function. So we didn't modify the loss function of GANs at all. We just passed these conditions using these modulating algorithms, and the model has learned the implicit correlation between the conditions and the data points. The other thing is that the generated realization respected the condition while maintained geological consistency, and this was measured by 2-point correlation functions, as we have seen before. The other thing is, although we've seen that the model has a very good interpolation performance, it had a limited extrapolation performance. So beyond a certain threshold, the model performance deteriorates and was able to generate something that's not correlated and not necessarily consistent geologically. But this is somehow expected from a machine learning model. It's known that the machine learning models are known to have poor extrapolation performance. So there are like several possibilities of continuation of this work. One of them is to like combine these different conditioning methods into one model. So to have like a one model that that's trained to perform soft conditioning, hard conditioning, or conditioning on global properties in just one training algorithm. The other thing is, which was also quite interesting for us, is to look at large scale generation. So all these data set that I have just shown are of limited resolutions, like 64 by 64. So one interesting question is, can we go beyond that in terms of resolution, in terms of the size of the generated images? So yeah, I think that's all what I wanted to share for this webinar. Thank you so much, and if you have any questions, I'm happy to answer them.
Thanks, Alhasan, for the very interesting talk. I first apologize for flashing other slides on top of your slides because we are having a technical difficulty with the next speaker slides. So the floor is now open for questions. So if you're watching us from YouTube, please sign in and post your questions, and then it would be forwarded to our streaming platform, and then we're going to read it. So Alhasan, you have worked on generative modeling for a while. When you started in the pre-ChatGPT era, where there was some knowledge about generative modeling, but now everybody knows about generative modeling, especially in natural language processing. So what can similarities you can, I understand that you are studying geology. We don't see geology in the subsurface everywhere. We have limited training data in comparison to all the texts which have been collected from Wikipedia or the, I think it's a Pile database, which is used for training all the large language models plus extra data sets in terms of terabytes of data. So what is the challenge and the differences between training on natural language processing and images and snapshots from YouTube videos versus your application?
Yeah, thanks. So it's a very interesting question. So in terms of training on natural language processing data, this is the main thing is that these data are available everywhere. You can look at GPT models or the models that have like LLM models. They can acquire data from everywhere. Wikipedia, GitHub, research papers, everything. So it's the availability of data is there. The only question for them is just they have the training power to train on these kind of large corpus of data. When it comes to my application, there are two issues here. The first thing is that the data itself might not be available. So we can be interested in a particular application, which doesn't mean that we have everything regarding this particular problem. The second issue is going to be the computational power. So just by looking at LLM models, they have tremendous GPU powers to train these large models. For us, it's not possible unless we're working with like very we have like the computational resources to do that. So I think this would be the two main issues.
Okay, we haven't received any comments, so I will lean on my co-chair, Menjia. Do you have any questions for Alhasan about generative modeling in general and his specific application of generative modeling in the subsurface?
Yeah, I just have a general comment. I'm just curious because you mentioned about the computational cost for large language models. So how about the ones that you are currently use? Like what's the most heavy part? Is it the training part or the generation part? Could you briefly comment on it? Thank you.
Thanks. So most of these data is just not very large. So I think the largest one that we worked with was like 15,000 samples. Each one of them is 64 by 64. So these were trained on a single GPU of 16 gigabytes of RAM, and I think the whole training process took like 8 hours or something. The generation could be like in a few seconds, so it's super fast, but the training is not computationally expensive compared, of course, to large language model training. I think we also in this project worked with a 3D data set. I haven't included here in the slides, but I would say that this was the most computationally demanding problem. I remember that we needed to have like 4 GPUs to train the 3D model, which took almost like 1 day of training.
Thank you.
Thank you. And the challenge here, I would follow up on training large models. And the engineering challenge of these training kind of like runs and knowing that you are using finite resources. You mentioned that you have access to 4 GPUs. Let's assume that you are working on a larger scale. What can you learn from people who are publishing results in natural language processing while they are running on 1,000 GPUs? And what kind of techniques you can utilize there based on that learning to advance your own research?
Yeah, so there are several ways to learn from them. For example, we can use efficient computational ways. For example, if we are dealing with a large problem that requires massive computational resources, we can use lower precision instead of having high precision. We can do something like gradient checkpointing, which somehow accumulates gradients across batch instances without having to calculate the gradient in every batch in a way that can fill up the GPU RAM. So there are definitely ways to learn from them, but again, I think the main issue going to be on how we scale from our the data set that we have, scale from the small data set to the large data set.
Okay, thank you for answering our questions. I'll check. We don't have any further questions from the YouTube, so we will move to the second speaker, Nelson Lubanga, Dr. Nelson Lubanga from the University of Aberdeen, if I'm correct, is a quantitative geneticist. His research includes integrating diverse data sets, including genomics, phenomics, environmental, and phenotypic data to improve the efficiency in plant breeding programs and accelerate genetics gains. His interests include optimization of plant breeding programs, developing algorithms, and using advanced genomics techniques to improve quantitative traits in crops. Currently, he is implementing genomic selection in Miscanthus breeding program. And if I'm correct now, he's not in Aberdeen, he's Aberystwyth University in Wales. So we have technical difficulties, so I will be sharing the slides for Nelson, and Nelson will be asking me to move forward and backward. So I will remove myself from here, and the floor is yours, Nelson, and please ask me to move forward or backward when needed.
Yeah. Thank you, Professor. Yeah, thanks, Professor, for the nice introduction. So my name is Nelson, as the Professor said, and I'll be explaining to you how we are implementing genomic prediction in the Miscanthus breeding program and how this what kind of tools in AI and machine learning we are using to accelerate our breeding program. Go to the next slide, please. So as a special introduction, so Miscanthus is a C4 perennial grass that takes about 3 years to mature. And we have two main types of Miscanthus. So the first one is Miscanthus sacroflorus, and the second one is Miscanthus sinensis. So these are the main species that we are interested to improve in Aberystwyth breeding program. So just as an introduction, genomic selection is the application of genomic prediction models to estimate the breeding values based on genome-wide markers. So these breeding values are the ones that are used to make decisions in a breeding program for selecting superior varieties to advance or to release to farmers. And how the process goes is, so we have a training set and we have the prediction set. So the training set has got the phenotypes. So the phenotypes are the data that you collect in the field. It could be yield, it could be quality, it could be disease resistance. And then the genotypes are the molecular markers. So these molecular markers are the DNA markers that are different between individuals, and there could be thousands of them. And then, so what we are interested in is to predict a different set of population that has only genotypes, that is molecular markers. In this aspect, so we are able to do predictions on a different set of population that has only genomic information only. And this is because we train the training population using a model, and once we are happy with the model that is giving us good prediction accuracy, then we are able to predict the second population using appropriate techniques, which I'll share in the next slide. So click it again, please. So a general genomic selection equation looks like this. So whereby we have the Y is the phenotype that you are trying to measure. Then you have the mean. Then we have what you call the marker effects, and then we have what you call the marker covariates in the middle. Then towards the end we have the error effect. So this is a basic genomic prediction model. So based on this is where we have different methods. So all these methods are based on the fact that we have P is greater than N, meaning that the molecular markers generated during genomic prediction are way more than the number of individuals. So you could have like 20,000 markers or even 100,000 markers, but but for the training population has got only about 100 individuals or 200 individuals. And so this raises a prediction problem which we need to solve. So we have different methods that are used to solve this problem in genomic prediction. So we have the penalized methods, which are mainly the mixed linear methods and are mainly the ridge regression, which are the shrinkage method, the lasso method, and also we have what you call the Giblat method, which falls under this category. And then we have the Bayesian methods. So the Bayesian methods we have are based on assumptions. So some of them assume that the marker effects are very small, others are very large, others assume that the marker effects are almost zero. So, and here we have different methods. So we have the Bayesian ridge regression, we have Bayesian A, Bayesian B, and Bayesian C, and also we have the Bayesian lasso methods. So, and then the last category we have artificial intelligence methods. So these artificial intelligence methods are differentiated from the first two, the penalized and the Bayesian method, by the fact that they are able to incorporate the additivity in the genomes. So while the first two mainly focus on the additivity of alleles, but the last one are able to incorporate the additivity of the alleles. And we have deep learning methods and machine learning methods, which are all categorized into supervised and unsupervised methods that can be implemented. Next slide, please. Click again, please. So how we measure how good a model is by use of cross-validation methods. The common method we use, it could be four-fold cross-validation. In this case, a four-fold cross-validation means you have divided your data into 4 sets. So if you have 100 individuals, you divide them into 4 equal parts. In this case, 20 equal individuals. So then you use the last method. You use one set as the testing set and the remaining sets as the training set. So you train a model based on 3 sets, and then you predict that model. You predict the remaining set using the training set, which was based on the 3 sets. So in that aspect, you're able to get prediction accuracy in each of the sets. So for instance, if you can click it again, please. So for instance, in this case, you have these 4 sets. So once the model is running, you train a model. So you first of all predict this you predict the first set based on the 3 sets, and then you're able to get the prediction accuracy, and the prediction accuracy is based on the predicted values, the correlation between the predicted values and the actual values. So based on what you predict and what was actually there, you're able to get a correlation between them, and then you record. Then towards the end, you do a mean correlation of all the, a mean value of all the predicted values in all the 4 sets. Next, please. So then I thought of introducing this term called the breeder's equation. So this breeder's equation helps us to measure the rate of genetic progress in a breeding program. So having a good model is one of the ways to increase the prediction accuracy or improve the accuracy of measuring traits in a breeding program. So this method has got what you call the intensity of selection, which is I. R is the selection accuracy, which in this case is the model that we use. And then the other one is the genetic diversity in the population, and then divided by t, which is time. So what we're interested in is to increase the selection intensity, also to increase the selection accuracy and the genetic diversity in the population, and reduce the cycle time. So for instance, for Miscanthus, it takes about 3 years to grow from seed to flowering. So typically, if you are to select a good variety, we have to wait until the third year before you can make any selection, because that's the long it takes. But with genomic selection, we're able to select at the seedling stage, because instead of waiting all that, you're able to have assuming you have the good model for selection or for training, you can, and then you have the population that you need to select from at the seedling stage. You can go and sequence, because sequencing nowadays is very cheap compared to phenotyping. You sequence, you use the training model that you had trained to do the prediction of the individuals that you are interested in a breeding program. And I'll demonstrate to you how we've done this in our breeding program. Next slide. So I thought of showing the phenotypes, how they're distributed in the population. So we had a trait called yield. So on the left-hand side is the Saclophorus population, which was more than the Sinensis population. So how we try to visualize the yield in this aspect is, on the x-axis we have the ancestry. In this case, is the maternal individuals, and on the y-axis is the number of how spread each of the values are. So, for instance, you could see that the 259 was the most spread compared to, let's say, 248 parental. And then on the right-hand side is the Sinensis population, which was very few compared to the Saclophorus population. So that's how I wanted to show how we visualize the two species. Next slide, please. So then for molecular markers, we had both the training population and the seedling population. So, and the two populations that we are interested to predict are the C0 population and the C1 population. So the training population had both the phenotypes and the genotypes. So the phenotypes are the trait, which is yield, and the genotypes are the molecular markers, in this case, DNA information. So we had about 41,000 DNA information, and then for prediction purposes, we had about 160,000 SNPs for the C0 population, and the C1 population, we had about 11,000 SNPs. But for genomic selection to be of use, we are interested in finding common markers between the training population and the prediction population. So I went a step ahead to determine what are the common markers between the training population and the seedling population. So for the first population, which is the C0 population, we had about 21,000 DNA information, or what I'll be calling SNPs. SNPs is a short form for single nucleotide polymorphisms. And for the C1 population, we had about 3,000 SNPs. So these are the ones that I used in the prediction of my breeding program, of the C1 and the C0 and the C1 populations. Next slide. So these are the common genomic selection models that I implemented. So I use a cross-validation method called C1 cross-validation, which is a 5-fold cross-validation method. And I, first of all, tried to check the predictive ability that were giving me, like, which was the best method I could use subsequently to select superior varieties in my program. So we have the GBLAB, we have the RRBLAB, Bayesian A, Bayesian B, Bayesian pi, and Bayesian lasso. I didn't use my machine learning method in this case because we are mainly interested in the additivity of the traits. So we were not interested in machine learning method, but that's what we are incorporating as the next step in our selection. Next slide. So you can see that. So on the orange box is the one species, which is a Cyclophorus, and the green box is the Sinensis. So we didn't find significant differences between the predictive ability within species, but we could see some differences between species. So the models gave higher predictive ability for the Cyclophorus population and lower predictive ability for the Sinensis. And this was attributed to the fact that we had a much larger training population for the Cyclophorus compared to the Sinensis. So the training set sizes also matters a lot, because when you have a bigger training set, it means that you have more individuals, and the model can be able to get more information from the training set compared to if you only have a few individuals to train your model. Next slide. So I thought of showing how a single environment, because we are interested in predicting genotypes in a single environment, and later on we are interested in predicting genotypes in multi-environment. So for the single environment, this is the model and that I described previously. And what we are assuming is that the genotypes and the markers are normally distributed with zero mean and variance. And of course, the error value is also normally distributed, the error variance. And how we determine the marker effects is that we have the genomic relationship matrix, and also we have the variance of the model. So we determine a covariance matrix between the genotypes. And in this case, we are using both the markers and the genotypes. And also, we also determine a covariance between the residual values. So we kind of model the incident matrix and the error variance in the model. So next slide, please. So the predictions we got by using this model, which is the GBLAP model, was in the first generation, which is the C0, you can see that the median value was a bit lower compared to the subsequent generation. So in this case, we could say that we made some improvement in terms of our prediction as we go along. So these are just 2 generations for the Cyclophorus. So we hope that next year when we make selections, the mean and the median values will continuously improve, and this is how we measure genetic progress in our breeding program. We aim to continuously increase the genetic progress by using better methods in our analysis. For the second species, you can see that we made a significant improvement in terms of the median values. So you can see that the second generations, because the first generation had quite low compared to the next generation. So, and we'll be keen to see what happens next year when we make selections and we apply genomic selection to help us do the selection to see if we are making genetic progress or not. Now, the next slide. So the next step is we have so far used a single environment model. So the next step is to incorporate, first of all, machine learning models. And secondly, is to incorporate what we call a G by E, or genotype by environment interaction in effect in the model. So in this case, the previous model only had the main effect of the genotype, the environment, and the markers. But now we are incorporating the main effects, so the interaction effects of genotype and environment interaction. And how we are doing this, we are building a covariance functions. First of all, we have different way of multiplying. If you've done a bit of matrix algebra, so we have what we call the Harderman product. Like this is the interaction here is called the Harderman product, whereby you are connecting the markers with the genotypes in different environments. So we have two covariance functions, one of the markers and the one with the different environments. And then we're trying to connect the individuals using those covariant functions. The other one is what we call the Kronecker product, where you are multiplying the markers in different in one matrix with the environment on the other matrix to connect those individuals. So that is what we are doing next. And also, we are going to use some of the machine learning models and possibly also bring some of the deep learning models to see if it's going to improve our prediction accuracy, or we can continue using our linear mixed models to inform decisions in our breeding program. Then next, I presume is the last slide. No. So lastly, we have a technique called sparse testing. So in our breeding program, we realize that when the plant breeders are planting genotypes in different environments, because they need to select varieties that are more stable across different environments. And it is expensive to, you can imagine if you have like 6 environments to make selections or to choose or to evaluate your genotypes. So if you have 100 genotypes or varieties, it means you have to plant all those 100 varieties in those 6 environments. So instead of doing that, we proposed a method called sparse testing. So this sparse testing is we are only testing a few varieties in each environment. And then using the model that I just described in my previous slide of using the covariance function of the model between the environments and the markers, we are able to predict the genotypes or the varieties that were not planted in each of the environments. So you only plant unique genotypes in each environment, then you use those few genotypes to predict the ones that were not present using only molecular markers and the marker covariates. Yeah, and then last slide. So this project has been a success based on a number of people who have present in this last slide, and we'll welcome any question if you have.
Thank you. Thank you so much for this very interesting presentation, Nelson. I was struggling in the back end because for some reason my slides were lagging by 1. I don't know. I see it behind. So we have the floor now open for questions. So please add questions in the chat in the YouTube. In the meantime, I would like to understand something. My understanding always that these kind of simulations that you have a large number of features in comparison to the number of samples.
Yes.
So how large is the feature space in your case?
So I'll give an example. So normally in this case, I had up to about 160,000 markers. I call them the molecular markers in this case. And the number of individuals are about 1,000 individuals. So you only have 1,000 individuals and 160,000 markers. So this one produces a modeling problem. So, and that's why we use what we call a lambda or Bayesian methods to kind of deal with that huge number of individuals. So with the lambda path, lambda is a penalization method, so that which allows including large number of covariates or marker covariates into a model when you have very few individuals to predict. So that's how we are handling the large number of markers or features, the way you're saying it, features compared to the number of individuals. So based on these models, the models that I described, all of them have got different assumptions that they make, so that you are able to differentiate between these models and to make informed decisions based on those specific assumptions, whether it's Bayesian statistics or machine learning or deep learning or the general mixed linear models.
And in terms of data set, is there any opportunity for transfer learning, or like, because I understand that the challenge of now a number of features is huge in comparison to the sample size.
Mm.
But can you learn something from other data sets and then build a foundational model or utilize a foundational model learned on open source, maybe some other application, and then adapt that model and refine it in your data sets at the end?
Yeah, thanks for the question. So the problem with this one is, one of the key requirements with genomic selection is that the training set should be closely related to the population you are going to predict. So in that aspect, if you use another population or another data set that is available in a different, let's say online or a different group, it might not give you the accurate predictive ability compared to a population that is closely related. So you are assuming, if you have individuals in 100 individuals, you make crosses or you make some cross pollination, you plant them, you are likely to get better prediction ability or accuracy compared to someone who will use a different population, let's say from a different country or a different environment, to do the same prediction. So that's the problem with this method. So the population has to be a bit closely related to each other than distance. Yeah.
Thank you.
Mm-hmm.
So we have a question from David. David was watching online on YouTube. He's saying thank you for a very interesting talk. There is some lag in the slides. Apologies for that from my side. So he might have missed this point, and he wants to know about how this study advances your field or improves the Miscanthus plant feeding process.
Yes, thanks for the question. So first of all, we are able to reduce the cycle time. So instead of waiting for 3 years to do our selections, we are able to make our selections at the first year, because we only require DNA information to make predictions, because we already have a good model to make predictions. So in that aspect, we're able to cut the generation interval by close to a third, for 2/3. We remove, like, instead of 3 years, we do it once. Then we are able to improve the prediction accuracy. That's the other aspect of this technique. So with phenotypic selection, assuming we just go and plant varieties in the field and then make random crosses, then we plant them, we all know that there's a lot of errors in the field. And just using the normal yield to make your breeding decision introduces a lot of error or biases in the selection. So with genomic selection, because you are using DNA information, so DNA information is very accurate. Your DNA is very different from someone's DNA, so there's no way the DNA can be wrong. So because you're introducing DNA in our selection, that makes the selection process more accurate compared to just using the physical measurements. And we know physical measurements are affected by the environment. We know the environmental influences, because even you, for instance, if you go and grow in Africa or you go to Asia, you have different environmental impact on you, and that could affect the yield of the traits, and it could actually make some of their selections not correct. So that's why we are advocating for use of molecular markers and a good model. It could be machine learning model, or it could be mixed learning model, or Bayesian model in our selection.
Fair enough. Thank you for answering that question. I have a small follow-up question before we conclude. You were showed a figure where is an an increase in the predicted dry mass between the generation. And for one one type of plant, it was about 15%, and the second one it increased a slightly small amount, and the second one it was 60%, and it increased to 65. But these results, if I correctly remember the caption there or the figure, the axis there, it was predicted.
Yes.
Not the measured. Is there any measured, or we need to wait for 2 more years to get the measured?
Yeah. If you have to wait for 3 more years, then you'll have to get the correct measurements. And so you see, in this case, we are interested in advancing our selections quickly. And because these models have been proven to work, and because based on our prediction accuracy of like 0.4, which was equally good, so we are kind of very happy with their predictions, and we could apply that practically in our breeding program. So if you wanted to see if we actually made good genetic progress compared to the phenotypic measurement, we'll have to wait for like 3 years, measure those traits, and then try to compare with what we selected. So basically what we'll do is to, again, measure them and then try to re-rank those varieties and see, okay, which ones did we select? Assuming we waited for 3 years, will you have made the same selections or not? And then the other thing is, you remember the point I made about relying on phenotypic measurements, because it's not accurate to just rely on phenotypic measurements. But because we'll have the molecular markers also there, we can still do our predictions based on the full data sets and including the markers to make the rankings. And that's why we can now compare, okay, did we make some progress or not? So it's a good method that most breeding companies, these big companies like Bayer, Syngenta, are using to advance their breeding decisions. And there's a lot of computational aspect involved in trying to train the models. So apart from just, to be honest, I've described a very basic model. We have other complicated models whereby you are also adding environmental covariates like weather data, like temperatures, like rainfall data, like soil conditions, different geographical covariates that you can measure. You can put all that in the model to improve your prediction accuracy. And also, nowadays we have what you call the phenomic. So instead of using DNA information, which is expensive, we are using what you call the phenomic information. So if you have near-infrared data sets, which is so much, if you are measuring near-infrared spectroscopy in one place, let's say it could be the leaf, and you have so many data points based on the wavelengths, so you can use that instead of using the molecular markers to make prediction in the model. So in that aspect, you're able to save your cost significantly, because near-infrared spectroscopy is very cheap compared to, let's say, sequencing and getting all those DNA information. And yeah, so there's so much technologies that we are working with computer scientists to kind of develop these kind of tools in our predictions, and also in what we call the high throughput phenotyping, to phenotype these traits, not just yield only, but so many other yields, and then we try to put all those yields together to make our breeding decisions. Yeah.
Thank you, Nelson, again for the very interesting talk and for answering all our questions in a comprehensive way. We thank Alhasan as well for his contribution. And now I would like to announce the next webinar on the Tuesday, the 3rd of December at 2 p.m. as usual, UK time GMT. And that session we will have Professor Konstantina will be from the University of Oxford, and will present a talk about harnessing machine learning and CFD for advancing hydrogen safety, predictive insight, and real-time solution. And also, in addition to this webinar series and the conference, which we will host in December at Exeter University, so please participate if you can. We are also organizing, as one of the projects funded by the UKRI call for AI for net zero, hackathon at the end of March at Harriet University in Edinburgh. The hackathon will be dated, I think, 26th to 28th of March. And the hackathon is a 3-day event where we invite PhD students and PDRA from across the UK to collaborate on AI-driven solution for carbon capture and storage. We will have 3 challenges, one about material discovery and properties of materials which haven't been manufactured yet, one about surrogate modeling for fluid simulation, the third one will be about policy and economics and natural language processing, how it helps us in analyzing different policies. So the participants will work alongside with researchers from the project to tackle semi-real world challenges. And there will be travel bursary. So please visit the website. I think the banner is wrong. The banner is here. It's called Hackathon 2025, and it is, if you search on Google, you will find it. So, and if you have any questions, please email me or email the webinar organizers. Thank you again for joining us, and we look forward to seeing you next week. Please spread the word and subscribe to the YouTube channel. And thanks again to my co-host and to the speakers of today. And bye now.
Bye-bye.